Yeah, go ahead.
Okay, thanks, Yash. Hello, everybody. I hope you can hear me loud and clear. Welcome to the session on Micromoths, brought to you by Moths of India website and a number of other partnering organizations, Titli Trust, I Found Butterflies, NCBS. Today's presentation is a subject that I know very little about. So I'm very eagerly looking forward to Jahir's presentation on micromoths. It's very interesting that he is at such a young age. I mean, Jahir is from Bangladesh. He's currently doing a BSc in Chittagong University. And at a very young age, I've been really impressed with the knowledge and the depth to which he has got into the world of moths. In fact, he's just come on board and he's supporting the Moths of India team as well. And he's been providing invaluable and very insightful information on a number of moths, including micros. Given the fact that for all of us, these tiny moths are beautiful, but yet not well known, I'll hand this over to Jahir and let him take the lead to tell us more about this. As I mentioned, we will take questions at the end, but if there are some very specific questions on a subject that Jahir is talking about, please put your questions in that box and we will ask him. Thanks, everybody, and over to you.
Okay, thank you, Sanjay sir, and good evening, everyone. Welcome to today's discussion on an introduction to micromoths, organized by the Moths of India team in collaboration with Titli Trust, Indian Foundation for Butterflies, and NCBS. I am Jahir Rayhan here today, and actually I am feeling scared and very nervous today because despite having experience in delivering speech on evolution, conservation, and taxonomy of moths on several occasions, I am feeling very nervous because what I am seeing is that everyone present here today, except me, has immense knowledge and expertise on various aspects of Lepidoptera, while I am a very beginner and a very amateur person. So before starting my discussion, I do request everyone to forgive me and correct me as well in case I make any mistakes at any point of my discussion. Because I do strongly believe that learning is a collaborative process and together we can learn this diverse group of insects. Okay, before starting today's discussion, let's clarify one thing, that who are actually micromoths? Whenever we created a group in Facebook named Microlepidoptera of Southeast Asia, everyone asked us that what are actually micromoths? Well, but the answer to this query is not straightforward because of several reasons. Firstly, the grouping is not monophyletic as well. I mean, the term Microlepidoptera and the groups we consider as Microlepidoptera is not actually a natural group. And secondly, some people think that size plays a key role here to distinguish micromoths from macros, but this is not true all the case. For example, there are several species of family Zyginidae, which is a Microlepidoptera family. The species are larger than the well-known Macrolepidopteran groups like Noctuidae family. So I think that it is better to clarify the distinction between Microlepidoptera and Macrolepidoptera. Well, actually, we consider in general term or in everyday use the Microlepidopteras as the following group. Everyone except this clade Macroheterocera, which is a monophyletic clade of in Lepidoptera, which includes Drepanioidea, Noctuoidea, Geometroidea, Laciocampioidea, and Bombicoidea group, and butterflies as well. So everything except these 6 groups are considered as Microlepidoptera in general term. So we will use this definition in case of our discussion. I think that it's clear now that which groups we are considering microlepidopterans. Wait, uh, before starting our today's discussion, I would like to remember my Gurudev, Niels Peter Christensen, who is considered one of the godfathers in case of taxonomy and evolution of Lepidoptera. And I do strongly believe that if he had not been born, then the taxonomy and evolution of Lepidoptera would have remained largely unresolved. So why I am showing him in this slide, there's a reason, because you are seeing here a very simplified phylogenetic tree of major groups of Lepidopterans. The order Lepidoptera is divided into 4 suborders or 4 groups, which are Geogloptera, Aglossata, Heterobathmia, and Glossata respectively. So while Niels Peter Christensen and his colleague Nielsen describing the new suborder Heterobathmina back in 1982, he commented one thing about the ancestry of Glossata and Heterobathmina groups. I think that I should clear one thing, that who are actually Glossata and who are Heterobathmina. Well, Glossata means a clade or group of Lepidopterans, the species of which possess coilable proboscis or tongue. And the other 3 groups, I mean Geogloptera, Aglossata, and Heterobathmina, they are considered as primitive moths, and they don't have any proboscis or coiling mouthparts. They instead have mandibles to feed on pollens of different species of plants. So while describing Heterobathmina suborder, Christensen commented as the ancestors of Glossata and Heterobathmina. You are seeing here that at this place, the ancestor which lived millions of years ago, he says that it may have a wingspan of about a centimeter, a metallic purple flowering mottled with golden or silvery scales, and with a distinct tornal spot, inhabited a temperate area, was primarily diurnal and early spring flyer, and had a larva which was devoid of prolegs and made blossom mines in deciduous trees of the order Phagales. And you already know that most primitive groups of moths, I mean most microlepidopteran families, show this behavior still now. I mean most of the groups have larvae which mine different species of plant families. Well, I think that this is a major confusing issue while dealing with micromoths, because we know that the Lepidopterans, I mean moths and butterflies, share their ancestry with Trichopterans or caddisflies. So because of this ancestral relationship with caddisflies, most primitive moths and micromoths appear to be in posture, size, shape as Trichopterans. So while dealing with micromoths, it's necessary to sort out Trichopterans and Lepidopterans. And the relationship which I am showing you, that Trichopterans and Lepidopterans are considered as belonging to a group because of their similar ancestry, is proved by so many characteristics. I mean, there are already 18 adult characteristics which unite Trichoptera and Lepidoptera along with several extinct groups of insects within a single group or clade, which is known as Lepidoptera. One of the characteristics, which is my personal favorite, is the wing venation, which is seen in Trichopterans and Lepidopterans, which is that in case of Trichopterans, I mean primitive Trichopterans, they have a very specific characteristics on their forewing venation, and that is their anal vein looks like double Y shape, you are seeing here. And this character is also found in some primitive Lepidopterans group as well, like you are seeing here wing venation of a primitive group of moths, which is also showing this double Y formation. And based on these similarities, scientists have made this analysis and have made these illustrations on hypothetical ancestral forewing venation of Lepidopterans, which is this image. So now question is how to distinguish between Trichopterans and Lepidopterans. Well, you are seeing here in this slide that I am showing several micromoths and trichopterans. So can anyone say that which is this insect? Is it a moth or lepidopteran? I mean, is it a moth or caddisfly? Anyone?
Type in the text box. Caddis fly. Yeah, I would agree too.
Okay. Can anyone say that what are these two insects? Are they moths or is there any caddisfly?
It's a trichoptera.
Okay, thank you. Yes, it's trichoptera. Okay, now next thing. What are these two insects? Is there any trichopterans here or both are lepidopterans?
I think both lepidopterans.
Oh, there's
But the one on the right is probably not. Yeah. Okay.
I think that the moth and lepidopterans both have been successful in deceiving us because there is one lepidopteran and one trichopteran here. The left one is lepidopteran while the right one is trichopteran. Okay, next pair. Is there any trichopterans here?
Roger says right side is trichopteran. So
yes, he is right.
And yes, both moths. I would have said both moths. Yeah.
Left one is moth while right one is trichopteran. So we are already seeing that it's often confusing to distinguish trichopterans from moths. So let's discuss on this issue that how can we differentiate moths from in trichopterans. So it's not actually easy always because some primitive groups of moths still show some characteristics which are very much similar to trichopterans. However, most major groups of moths can be sorted out from trichopterans based on their mouthpart characteristics. You are seeing here that the left image belongs to the mouthpart of trichopterans, while the right side is a mouthpart of lepidopteran. And you are seeing that trichopterans have characteristics maxillary pulp, which consists of 5 segments you are seeing here. Though some primitive moth groups, for example, the most primitive moth groups, Micromoths, also have these 5 segmented maxillary pulps. And usually trichopterans keep their maxillary pulps and labial pulps in a ring-like structure. I mean, you are seeing that maxillary pulps are keeping forward to their mouthpart, hostelum, and labial pulps are keeping backward to their hostelum. So it makes us somewhat ring-like structure. But in case of lepidopterans, their maxillary pulps is usually reduced. Though I have already said that some primitive groups like Micromoths still have maxillary pulps, which is very much like trichopterans. And seeing that there are some important features in their legs which can be used to distinguish lepidopterans from trichopterans. You are seeing that left side is showing the legs of trichopterans, while right image is showing the legs of lepidopterans, and you are seeing that the trichopterans legs don't have any scales, while lepidopterans legs have scales. And secondly, we all have observed grasshoppers so many times in field, and we know that grasshoppers have serrated spine-like structures on their leg, like this one. And these characteristics is also found in trichopterans. I mean, their legs are covered with numerous tiny spines, while lepidopterans don't have this much spine. Instead, they have a very little amount of spines. I mean, a limited number of spines or spurs on their legs. So I think that we all have got some idea to distinguish lepidopterans from trichopterans. However, the most important feature to differentiate between them is the presence and absence of scales on their wings. We all know that trichopterans doesn't have any scales on their wings. Instead, they have hairs. On the other case, lepidopterans have scales on the wings, and and on all over their body. So this is the summary of this slide, that how to differentiate Trichopterans from Lepidopterans. We can make it possible, and their maxillary pulse, their legs, and characteristics mouth part. Okay, frankly speaking, it is very tough or sometimes impossible to identify micromoths, at least up to family or subfamily level, during field or from photographs only, because most of their identifying features are so subtle and so minute that it often needs to analyze them carefully under microscope. However, following these things, we can at least get any idea that about superfamily or family level identification of Microlepidoptera, and that are posture, head, labial pulse, etc. You are seeing here, and I will discuss more about these things in my next slides. And I am showing you a very brief guide to photography, because whenever people show us their photographs to identify their moths, it's often difficult for for people like us, because we often need images of good quality and good resolution and of different angles, because we need to see different characteristics to identify their families. So I think that considering these two guidelines, we can at least identify our micromoths up to family level. And that whenever you will take photographs, please try to take close-up photographs of different views with good resolution, at least dorsal and lateral image. And secondly, carefully observe and note down different styles and behaviors observed at the field, because many moth groups show twirling behavior, some are erratic in flying, etc. And those behaviors can aid in identifying them at least up to family or subfamily level. Well, for today's discussion, I have included these families in my slides, and I have excluded some families, some well-known groups of moths like Coccidae, Cecidae, Hepialidae, Pyraloidea, etc. And I think that they are easy to identify at least up to family level. So this is why I have excluded those families and included these families for today's discussion. And I am grateful to these persons for allowing me to use their images in my slides. So let's start our journey with families of micromoths. The first family, the Micropterigidae. This family is considered the most primitive groups of moths, and this family is actually widely distributed from Old World to New World. However, from India, only 2 species are known, and they are mainly based on some very old collections of museum specimens, which were made by late Mr. Atkinson and Maria Eastchili. And late in 2010, Niels Peter Christensen and his colleagues described these 2 species which were collected from India. One is Micropteryx cornuela, which is known from Mishmi Hills, Arunachal Pradesh. You are seeing here the images of the species, the male and female respectively. And the another one is Micropteryx longicornuela, which is known from Darjeeling. But the interesting fact is that after their description or after the collection made by British people, we still don't have any observation or any data pertaining to this species or even pertaining to this family from India. So I think that we amateur persons who make field visits or field observations, we can search for these families in from India and from adjacent parts like Bangladesh and other countries. And this family is actually characterized by their functional mandibles, which is used to feed on pollens, and their wing venation, their maxillary pulps. You are seeing here that their maxillary pulps looks almost identical to those of Trichopterans, but they can be sorted out from Trichopterans seeing their scales on the forewing. And another feature which is seen during their resting, it is that whenever they rest, they keep their antennae forward and making almost 45-degree angles to the surface. You are seeing here that their antennae are kept keeping almost 45-degree angles to the surface. So I think that following these characteristics, we can identify these families. And the second family is family Paleosetidae, which belongs to superfamily Hepialoidea. And the family name Paleosetidae means paleo means archaic, while setidae means city or hairs. I mean, the family actually means that hairy archaic moths. You are seeing here that these two moths belonging to this family, which was observed by Ashok Sengupta from India, and I think that this is the very first observation of this family in live habitats from India. And this family is actually characterized by their hairy appearances, and as they belong to superfamily Hepialoidea, they also have very small antennae, which is found in other families of this superfamily like Hepialidae. And their proboscis is actually absent, and we already have said that their antennae is very short. However, there is a very important feature in the male genitalia of this species, which is that their valves, which is I am showing here, that their valves of their male genitalia consists many characteristic bristles. You are seeing here. And following these characters, I think that we can identify this family from field level or by observing them in museum collection. Okay, I should mention one thing, that only two species of this family are known from India. Both belongs to genus Xenastis, and the species are Xenastis minutus and Xenastis lutata respectively. And interesting fact is that both species are known from North Indian parts. And next group is superfamily Nepticuloidea, which contains two well-known groups of moths, family Nepticulidae and family Opostasidae as well. And whenever we need to identify this group, we can see the several characteristics. For example, they are very small sized moths, and their wingspan can be range from 1.8 millimeter to 10 millimeter. You are already understanding that they are very small moths, and this is why some people call them pygmy moths. And they actually have wings as lanceolate shape, you are seeing here, and they are characterized by their scaly head and characteristic eye cap, which is made by the base of their antennae. You are seeing here that they have characteristics eye cap, and they have frontal tuft on their head. Okay, this superfamily consists of two families. We already have learned about this, the family Nepticulidae and Opostasidae. Now question is how to differentiate these two families. Okay, this is not easy always, because we all know that moths like to deceive us based on their appearance. However, following two categories, one is their wing color and second one is their wing venation, we can differentiate these two families. The moths belonging to family Nepticulidae usually have darker colored wings and often have a medial band on their wing. You are seeing here that this moth is darker colored and having medial band on their forewing. And on the opposite hand, the family Opostasidae tends to be usually whitish in color. And secondly, the important feature is their wing venation. In case of Nepticulidae, they don't have they usually have branched veins on their forewing. You are seeing that their forewing veins are showing branching patterns, while in case of Opostasidae, they actually don't have any branching vein on their forewing. You are seeing here that all veins are free. There is no branches on any of these veins. So based on these two characteristics, we can identify these two families or at least we can get an idea to about these two families. Now the next group is family Adelidae, which belongs to superfamily Adeloidea. Okay. The family name Adelidae is derived from Latin adelos, which means unseen. This is because of their larval habit of concealing inside portable case. You are seeing here that their caterpillars is concealing inside a portable case. And the adult moths are usually small, almost 23 millimeter in size, and they are usually glossy, metallic patterned or iridescent winged, and they have very long antennae. You are seeing here that their antennae is very long, and in case of males, the antennae are larger than the females, and they have some other characteristics as well, like their head is scaly and their eyes are larger. I will show you in my next slide. And they are usually day flying, and some species of this family, the female members of which may have basal hub of antennae with prominent hairs. You are seeing here that a female moth which has antennae which is showing that the basal part of the antennae is hairy. You are seeing here. Now, the next group of moths is Coreutidae family, which belongs to superfamily Coreutoidea. The family name has been derived from Latin coreutes, which means dancer, because the adults are actually erratic flyer and they can make quick and jerky movements. And the adult characteristics can be said like this: that their wings are usually stout and square shaped, and they are often metallic patterned, and their head are smoothly scaled, and labial bulbs are usually ascending. And I think that considering these three genera, we can identify this family better than following their characteristics, because these three genera are usually very common in India and adjacent countries, and that are genus Branthia, Saptha, and Coreutis. You are seeing that genus Branthia, the species of this genus shows some mimicry pattern with spiders. On the other case, the species belonging to genus Saptha shows very much similarity with Adelidae and some other families. And the species belonging to genus Coreutis have actually the ideal stout squared wings. We have already said that squared wings, which is a characteristics for this family. Now the problem is differentiating Saptha and Adelidae as well as some zygonid moths. Okay.
Jahir, just a question for Adelidae. How many species in India, and for the other next family also, how many species, just order of magnitude?
Okay, actually from India, one genus is known belonging to family Nepticulidae, which is genus Stigmella, so far as I can remember.
No, but from Adelidae, how many species would there be there in India? The order of magnitude, I'm just not asking for the exact number, and for this family as well.
I think that this family is very poorly studied from Indian subcontinent, so we can't make any assumption for this family. But however, please let me search in some literature that how is their global status.
No, go ahead. Don't worry. Yeah, we can come back to this later.
Okay.
Go ahead.
So my question to everyone that we are seeing.
Roger said tens and maybe 50 plus. So that gives us an order of magnitude. Yeah.
Okay. Thank you, Roger sir. Okay. The family Nepticulidae globally contains 13 genera and 819 species, while family Opisthozidae contains 7 genera and 192 family species globally.
Okay.
Okay. Okay. Can anyone say how many family representatives are here in this slide?
4.
4.
Two.
Well, anyone else?
I
3.
see zygonids and adelids.
Okay.
I think there's also, I think brachioid is also here, no? Okay, 3.
Oh yeah, there's a third one.
Okay. So we have 3 family members. I am showing you. They are Choriotidae,
Adelidae.
Zygonidae, and Adelidae. Now, question is how to identify them that correctly. So this can be possible just analyzing their head structure. You are seeing here. Okay. You are seeing that we already have discussed that in case of Choriotidae, they usually have ascending labial pulp. You are seeing here. The labial pulp is ascending and they have larger eyes. You are seeing here that their eyes are larger. And the most important feature is their head is smoothly scaled. You are seeing here, smoothly scaled head. Now, in case of Zygonidae, the most important feature to describe this family is the presence of giant chaetosemata on their head. You are seeing here that they have chaetosemata, which is very good characteristics to identify this family. And in case of Adelidae, their eyes are larger, just like Choriotids. However, their head is hairy. You are seeing here that their head contains many hairs on every side. So I think that following these characteristics of their head, we can differentiate Choriotidae, Zygonidae, and Adelidae. Now, next group of moths, the superfamily Tinioidea, which contains Tinidae, Eriocotidae, and Psychidae family. Now, these 3 families often confuses us because it's often challenging to differentiate Eriocotidae from Psychidae. However, I am showing some easy features which can be used to at least get an initial idea on their identity. Now, the first family, Tinidae, which is derived from the word tinia, which means destructive worm. We already know that Tinidae, the larvae of this family destroy cloths and textile goods. However, I think that they are very cute moths. The family is characterized by rough and erect scales on their head. You are seeing here that their head contains very rough and erect scales. And secondly, they often have inconspicuous maxillary pulps, which can't be always visible from photographs. And they have a very noticeable characters, which is their larva contains characteristics larval case. You are seeing here, which is seen very often in our house. And second family is Eriocotidae. You are seeing here Eriocotidae family, which is characterized usually by their bipectinate antennae, while tinids usually have monopectinate antennae. You are seeing here that their antennae is thread-like, not feather-like as in Eriocotidae. However, the family Eriocotidae is best defined by their wing venation and genitalia characteristics, and notably the presence of microtrichia scattered over all wing surface, but the characteristics is not visible from photographs. We need to analyze set up specimens. However, I personally identify this family following these characteristics on their wing. You are seeing that this portion of their wing shows something, that you are seeing that this V-shaped structure, is it visible? This V-shaped structure, which is formed by the folds of their wings. This feature can be used to identify this family, I think, because most Eriocotidae members, which I have observed, contains this feature. So I think that this feature can be used to sort out this family from photographs. Now, next family is Psychidae.
Zoya, there's a question from somebody saying, how can we separate male and female, if at all?
Okay, actually, if they show sexual dimorphism, then it can be easy to distinguish males from females. If they have very prominent dimorphism, but if they don't have any sexual dimorphism, then it's often tough to separate males from females based on photographs only. However, in case of Psychidae, their females can be separated from males because their females usually tends to be brachypterous. They don't have any functional wings, so they remains inside their characteristics larval case. And males are flyer, and females emit pheromone to attract males, and males travel long distance to mate with the females. And this is why, to increase surface area, the males tend to have branching pattern on their antennae. They have bipectinate antennae, you are seeing. Okay. So the family Psychidae, the name derives from the word psyche, which means the soul. The word actually have a very interesting meaning, because it said that being the spirit of man liberated from the impurities of the flesh. And this line refers to the fact that some members of the family Psychidae reproduce by parthenogenesis without male involvement. So now question is how to identify this family. This family is very easy to identify from caterpillar or larval stage, because their larva has characteristics larval case. You are seeing here that their larva makes case using twigs or other environment, other things. You are seeing that I am showing 3 larval case which occurs in Bangladesh. One from the top is very much like pagoda, and this is why it belongs to genus Pagodiella. And the lower left one, which has a spiral pattern, it belongs to genus Mohasena. And the right one, having twigs and some twigs are elongated, this is the feature of genus Eumeta. And the adult of this family moths usually have wings with darker colored or smoky colored, and they can be sometimes transparent winged, and their antennae are usually pectinate. And you are seeing here that 2 examples of this family. One is Eumeta variegata and another one is Degia. And I think that if you notice one thing, at this point of their wing, you can sort out or you can distinguish that this does not belong to family Eriocotidae but Psychidae instead, because I have already said that in case of Eriocotidae, this part of their wing usually show V-like structure, which is formed by their folding of wings. However, family Psychidae members doesn't have this V-like structure. So this is easy to separate a Psychidae from family Eriocotidae. Now, I think that if we follow this 3 literature, which is the Fauna of British India Volume 1, and another one which was published by G.M. Das back in 1956 on Psychidae of Northeast India, and another one is most importantly the works of Mr. Dyer, who is an expert on this family, and he has numerous publications pertaining to this family in ResearchGate and other sites. So following these 3 literature, our Bangladeshi moths have been identified, and we have identified 5 species from Bangladesh. They are Eumeta variegata and Mohasena species, Pagodiella species, Degia species, and Amatissa species. And these 5 representatives of this family occurs in Bangladesh. Now the next family, which is Gracilariidae, which belongs to superfamily Gracilariidae. The family name has been derived from the word gracilis, which means slender, meager, or plain, generally in a pejorative sense. You are seeing that they are very slender moths, and their characteristics, most important characteristics of this family is their resting posture. You are seeing that wherever they rest, they usually keep their body, the front part of their body elevated. You are seeing that this moth has kept its front part of the body elevated. And if we examine it closely, we can see some notable features on its head, which also can be used to identify this family. And that is, they have curved labial pulp. You are seeing here that these moths have curved labial pulp, though this characteristics is not apomorphic for this family. I mean, these characteristics is also found in several other groups of Lepidopterans. And you are seeing that their heads are usually scaly or hairy. You are seeing large hairs on their head. And following these characteristics and importantly their resting posture, I think that this family can be identified as at least we can say up to family level. However, genus and species identification is challenging. And the caterpillars of this family are usually leaf miners, which is a very notable feature of Microlepidopterans. Now the next family, Plutelidae, which belongs to superfamily Eponomeutoidea. The superfamily name Eponomeutoidea is derived from the word Eponomeuta, which means making underground mines, because most species of this superfamily have caterpillars which usually mine leaves and shoots. And I think that this family is very well studied, at least one species, which is Plutella xylostella. I think that in literature which deals with pest species, the most importantly they discuss on this species, which is Plutella xylostella, which is a very notorious pest of plants belonging to family Brassicaceae, and I have given some insight into its life history. And you are seeing that they have a very characteristic behavior at their larval stage, and that is their caterpillars or larvae usually make this type of holes or windows on the leaves of their host plant. You are seeing here that their caterpillars are making holes or windows on the host plant leaves. And the adult moths can be identified following characteristics lies on their head. You are seeing that they also have somewhat ascending labial pulp. You are seeing here that their labial pulp is ascending, and the most important feature lies on the second segment of their labial pulp, which contains triangular scale tuft. You are seeing here that the second segment of labial pulps contains triangular scale tuft, which is a very good indication for this family. And one important thing is, whenever they rest, they usually keep their antennae forward, which appears as like a horn or something else. So I think that we can keep in mind that Plutella xylostella, this species can be used as model to identify this family, because it's very easy to identify this species following its diamond-shaped pattern on the on its wing. Seeing here 1, 2, 3, and several diamond-shaped structures on its wing. So following this structure, we can easily identify this species. And if we consider it as our model, then we can identify other species of this family. Now, next group, the most species-rich and most diverse groups of Microlepidoptera, the great Gelechioidea superfamily. This superfamily has a very ups and downs in the history of their taxonomy. And recently, back in 2020, Wang and Lee have revised this superfamily, and based on their finding, now this superfamily is characterized by 20 families. However, I have included Lecithoseridae, Oikophoridae, Paleophoridae, Strathmoporidae, Schiztrididae, Cosmopterizidae, and Galechiidae for today's discussion. So if anyone wants to know more about this superfamily and their status, they can go through the paper of Wang and Lee, which was published in 2020. I have attached the reference of that paper. Now, this superfamily is characterized by their very characteristics labial pulp, which is curved and elongated labial pulp. You are seeing here that which is easily recognizable. And this labial pulp is whenever if you see any moths contain this labial pulp, then we can be sure that it certainly belongs to superfamily Galechiidae. Now, which family they are in, it's a very challenging issue because it's often very tough or impossible to identify the moth up to family level. However, let's try to discuss on some easily recognizable families of this superfamily. The first family Lecithoseridae. This family can be identified during field level following their resting posture and their antennae characteristics. You are seeing that the small sized moths usually have antennae which are larger than their wings, and the antennae are usually kept forward. You are seeing here that they are keeping their antennae forward. And secondly, their caterpillars are very important because they feed on non-living material and organisms like dead leaves. However, the family is best defined by their genitalia characteristics, which is not visible from photographs, and we need to examine the setup specimens. But following these two things, I mean their posture and their antennae, I think that we can identify this family easily from photographs. Now, the second family Oikophoridae, the family name is derived from two Latin terms, the term oikos, which means a house, and phorio, which means to carry. It is actually an inappropriate naming because the name is showing that their caterpillars possibly carry house or case as like Psychidae or other families. However, their caterpillars spin silken webs on or within leaves or stems of living plants under decaying bark or dead and decaying leaves. And some feed on dry vegetables, birds' nest, or even clothes. So none of them are actually house bearers. So this is an inappropriate naming, I think. However, we have nothing to do because this was named by early workers. So let's try to discuss on their characteristics. They are very small sized moths with scaled proboscis, which is not always visible from photographs, and they have smoothly scaled head. You are seeing here that their heads are smoothly scaled or sometimes rough on vertex. So to understand these features, we need good quality images, which I have already discussed. And their labial pulps are usually recurved, long, slender, and retracted. This is very important thing. However, we can identify this family following their resting posture, which is that they usually have very strongly tectiform or involute resting posture. You are seeing here that their resting posture, and their forewings are often broad or narrower with wing tornal angle. You are seeing that this moth seems to have no tornus at all. You are seeing that the outer margin is similar to the inner margin, and we are seeing no distinctive tornus in this moth. And from these characteristics, that we can at least get an initial idea on this family. Now, next family Paleophoridae. It has a very long history on its taxonomy. because sometimes it was considered as a subfamily and sometimes as a separate family. For example, once this group was considered as a subfamily belonging to family Oikophoridae in Hoses 1974. Later, Minet 1986 and 1999 made this subfamily a separate family Paleopodidae. And next, Haikila 2014 again made it a subfamily of family Depressariidae. And later, in 2020, Wang and Lee, whenever revising this superfamily at molecular level, they described this as a separate family belonging to the superfamily Garechioidea. So now the fate of this group is as family level following Wang and Lee 2020. Now the question is, how can we identify this family? Well, this family is well defined by their wing venation and their thoracic characteristics. However, this family contains at least two subfamilies, which are subfamily Acriini and subfamily Oditini. And I think that if we try to identify these two subfamilies and if we try to understand their characteristics, we can identify this family easily. Because in case of subfamily Acriini, the members belonging to that group tends to have emarginated wing costa on their forewing. You are seeing here that they are showing cup-like depression or emarginated wing. And in case of subfamily Oditini, they can be identified following their wing markings. And that is, they usually have spots on and around their forewing discal cell, as well as their as well as they have a submarginal line made of series of spots. You are seeing here. And following these characteristics, if we can identify these two subfamily, it's easy to identify this family, I think. Now the next family Stathmopodidae, which is derived from the word stathmos. It is a Greek word which means to stand or station, and the word podos, which means legs. This name means that adult moths, whenever rest, most often keeps one of their legs elevated. Though this characteristic is not contained within this family only, because this characteristics is in many other Lepidopteran groups. However, the adults of this family members can be identified following their characteristic labial pulse, which is most of the species belonging to this family have characteristics cowhorn-like labial pulp. You are seeing here that their labial pulps look as cowhorn, or at least their labial pulps are very slender and very long. You are seeing here that their labial pulps may be as cowhorn or maybe slender. You are seeing here. This family consists of three subfamilies: Stathmopodini, Cuprinini, Atkinsonini as well. However, whenever Wang and Lee revised this family and their phylogenetic relationship, the subfamily Atkinsonini behaved very. The behavior of Atkinsonini was confusing because the species belonging to genus Atkinsonia seems to be nested within genus Stathmopoda. So this means that this family is monophyletic, there's no doubt. However, the relationship between the subfamily is confusing. So this is why the three subfamily I have already mentioned is actually confusing and is problematic and is still unresolved. Now next family, family Schiztridae. The family name has been derived from Latin word scuthros, which means sullen. It is possibly because the term is linked to the teardrop-shaped appearance of the adults. You are seeing here an illustration which is giving an insight on their appearance. They always often appear to be as teardrop shape you are seeing here, and their heads are relatively smaller in compared to their body. And as they belong to superfamily Gelechioidea, they also have curved labial palps, and the species of this family also keeps one of their legs elevated while resting, as we have already said while discussing on Stathmopodidae, that this resting posture, I mean keeping legs elevated, is not confined within Stathmopodidae only. And this species, which I am showing you in this image, the Eretmosera impactella, it is a very well-known representative of this family from Indian subcontinent. So now question is, whenever we observe this family and other Gelechioidea superfamily members at field level, we often become confused based on their appearance, because you are seeing here that this moth and this moths both are showing elevated legs, and secondly both are looking somewhat similar in appearance. Now question is, do they belong to a single family or separate families? Well, now you are seeing that the second moth I am showing here belongs to family Stathmopodidae, while this one belongs to family Schiztrididae. Now question is how to differentiate Schiztrididae from Stathmopodidae, because we are seeing that both family members can show elevated legs and can show somewhat similar pattern and can show somewhat similar resting posture. Now we can differentiate these two families based on their characteristics labial pulp. You are seeing that in case of Schiztrididae, their labial pulps hardly exceed the level of their head. You are seeing that this is their level of their head, and this is the limit of their labial pulp, which is showing that their labial pulp hardly exceed the level of their head. While in case of Stathmopodidae, their labial pulps usually are very long and exceeds the level of their head. However, this type of characteristics, I mean, which I am showing here for Stathmopodidae, is also found in another family of superfamily Gelechioidea, which is Cosmoptericidae. So this makes confusion often to separate Stathmopodidae from Cosmoptericidae. However, we can separate Schiztrididae from Stathmopodidae and Cosmoptericidae based on this labial pulp. If it is small and if it does not exceed the level of head, then it belongs to Schiztrididae. But if it exceeds the level of their head, then it belongs to either Stathmopodidae or Cosmoptericidae. Now the next family Cosmoptericidae, on which I have already discussed some aspects. So this family name actually means ornamented winged. You are seeing here that this moth is showing ornamentation on their wing pattern, and I think that among the species which belongs to superfamily Gelechioidea, most beautiful species belongs to this family, because they have very beautifully patterned forewings. And you are seeing that I am showing you some. Okay, I think that someone has been unmuted. Can you mute yourself, please? And I think that someone is controlling the screen, because these lines or markings are not my lines. Someone else is making this pattern. So can you mute yourself, please?
Yes, you can disable this since you're the host. Just make it not possible for uh, uh, other users to do this. You can disable that. Probably somebody's kid is playing with the screen.
Okay, will. Okay. Let's discuss again. Okay, the adults belonging to this family usually have very long and narrow wings. You are seeing here, and their wings can be smoothly or sometimes having scale tufts. You are seeing here that this medial lines is made of scale tuft, or sometimes they may have patterns on their wings, or they may be brightly colored or metallic patterned moths. And they usually have very slender and long labial pulse. You are seeing here that they have very long and slender labial pulse, which often looks like scythe. And the one of the most interesting characteristic of this family is most species tends to have red eyes. You are seeing here this moth has red eye. If you find any red-eyed moth having long and narrow wings and having long and slender labial pulse, then we can be sure that it belongs to family Cosmoptericidae. Now, next family Gelecidae. The family name is derived from two Latin words, the word ge, which means earth, lekos, which means a bed. The word actually means sleep on the ground. This is from the fact that most of the species in the original or more genera feed on low growing plants as caterpillar, and as adult they usually tend to fly close to the ground. And for this reason the moths have showing these characteristics are considered to be a part of family Gelecidae. The question is how to identify these family members. The most notable feature of this family is hind wing, which is the apex of their hind wing is usually falcate or at least pointed. You are seeing, but I think that this is not often possible to see these characteristics from photographs or during levels. So how to identify this family from photographs? Now we can consider some things which are found on their labial pulp and on their head, and that is the members of this family tends to have smooth scaled head. You are seeing here that their heads are usually smoothly scaled, and the most important feature is the second segment of their labial pulp can be smoothly or roughly scaled. You are seeing here that in this example, the second segment is smoothly scaled. And in this example, the second segment is roughly scaled. And in this example, the second segment is roughly scaled. So if you find any Gelechioidea member having this feature, then you can be pretty sure that it belongs to family Gelechioidea. Now, which genus or species therein, it's a challenging issue. However, this family is best defined by a row of narrow anteriorly directed scales on the forewing radial vein, which is not actually visible from photographs or at field level. So from photographs, we can identify this family following the characteristic of their head and labial pulse. And one important thing is they usually have their antennae filiform. You are seeing here they have filiform antennae. Now, these were all for superfamily Galactioidea. Let's discuss on superfamily Pterophoroidea, which is consists of two families, family Pterophoridae and family Macropyridae. I think that among the microlepidopteran groups, identifying this family is very easy because the moths at their adult stage show a very interesting resting posture, which is they often have a resting posture which looks like the cross of Jesus. You are seeing here that their resting posture looks almost like the cross of Jesus. And this is why one of my friends called these moths as Jesus moths. Now, the and most important feature is that the family name Pterophoridae means feather carrier. So what does it mean? It means whenever we observe their wings carefully, we see that their wings are actually feathery. You are seeing here that their forewings have 2 feathers, you are seeing here, and while their hind wings usually have 3 feathers. So because of these feathery wings, they are called Pterophoridae. Now, the next family, Macropyridae. As I have already said, that Pterophoridae members show feathers on their wings, but this is not true for all the case. There are some exceptions as well. For example, the members of genus Agdistis doesn't have these feathers on their wings, though they belong to family Pterophoridae. Now, for this reason, the members of the family Macropyridae are often confused with family Pterophoridae. And you are seeing here that I am showing you a member of family Macropyridae, which is a very well-known species. I think that this is the only representative of this family from India, and this is Agdistopis singhala, which is known from actually South India and Sri Lanka and North India as well. So this you are seeing that Agdistopis singhala, which belongs to family Macropyridae, also somewhat look very close to their appearance to the family Pterophoridae. So now question is how to differentiate these two families. I think that we do not need to learn this thing because we only have one species belonging to family Macropyridae from India. So this is the species, and if we find anything like this, then that is Agdistopis singhala, or anything else which does not look like this belongs to family Pterophoridae. I think that this is the easy thing to identify these two families. However, I have written down several aspects on their characteristics, which are of very minute and needs to examine carefully under microscope. This is why I am keeping those characteristics. Now, next family, it is also easy to identify, and this is family Eleucetidae, which belongs to superfamily Eleutheria. The family name actually has been derived from the term Eleucita, which means gnats or crane flies. I don't know what's the connection between gnats and crane flies with these moths. However, like Pterophoridae, these moths also have plumes or feathers on their wings. But in case of Pterophoridae, the number of plumes are limited, as we have already discussed that their forewings usually have 2 plumes, while their hindwings have 3 plumes. But in case of Eleucetidae, they have so many plumes on their wings. You are seeing here that their forewings and hindwings are consist of plumes. The number is more than 3 and 2, and they usually have 6 plumes on their forewings and 6 plumes on their hindwings, though this number can be above. So anything looking like these moths, I mean having many plumes on their wings, belongs to family Eleutheridae. Now, next family, family Torricidae, which belongs to superfamily Torticoidea. I think that I don't know how to discuss on this family because I am not an expert at all on this family, and we already have our expert Shashank Pathur sir between us. So please, sir, forgive me if I make any mistakes to this family. So the family name Tortricidae has been derived from the word tortrix, which means a twister, because the caterpillars twist and spin the leaves of their host plant, and this is why they are often called roller moths. You are seeing here that I am showing one rolled leaf, which is actually a behavior of this family. Though this behavior, twisting or rolling up leaves, is not confined within the family only, because this characteristic is also found in some other groups of moths. Now, let's try to understand this family and its characteristic, that how to identify this family. Okay. Most notable feature of this family, which in cladistics terminology is called apomorphy, I mean the character which they have got from their common ancestor, is the characteristic ovipositor on their female. Though this characteristic is not seen from photographs and during field level, so let's skip this character and try to understand from other characters. Now, I think that if we try to identify the subfamilies of this family, then it is easy to identify this family, because the identifying subfamilies is easy. This family actually consists of three subfamilies, which are Tortricini, Olethriutini, and Clidanotini. Now, in case of Tortricini, the members are usually bell-shaped or having flat resting posture. You are seeing here two representatives of this subfamily, which have a somewhat bell-shaped appearance and flat resting posture. And they usually have a triangular marking on their forewing apical region. You are seeing here that in this moth, the marking is very prominent, while in this moth, the marking is not much prominent, but it's present there. So if we find any moth looking like somewhat bell-shaped and flat resting posture, and have this triangular shaped marking on forewing, and having crambidy-like labial pulp, then we can be sure that it belongs to subfamily Tortricini. Now, next two subfamilies, subfamily Olethriutini and Clidanotini. Identifying these two subfamilies are not easy at all, because we need to understand several things, and we need to spend many hours at field and before books and all literature to make a distinction between two subfamilies. However, these two subfamilies can easily be identified or be differentiated from other groups of Tortricidae and other groups of Lepidopterans based on their characteristic markings, which is found on their forewing costal margin. You are seeing here that these moths have some notable marking on their costal region, and these moths also have marking. These moths also have marking, and these moths also have this marking. And this characteristic is called costal stribuli in entomological term, and this characteristic is usually occur in subfamily Olethriutini and Clidanotini. So whenever we find a moth having this characteristic, I mean costal stribuli, then we can be sure that it belongs to either subfamily Olethriutini or subfamily Clidanotini. Though there are some exceptions. For example, Ceracea species. you are seeing here an example of that genus, Cerase stipatana, which occurs in India. These moths, statistically, though they belong to subfamily Titli. So we need better understanding, and we need to spend many times, and we need many experience to deal with the subfamilies, I think. Now my question to everyone, that who is this medial one? A moth or something else? Does it belong to subfamily Olethriotini or Clidanotini, or it's something else? Can anyone say this? Is there anyone? What is this? Is it a Tortricity family member or not? Anyone with any answer?
I would guess Tortricidae, but I'm not sure.
Okay. Anyone else?
It doesn't look like a moth to me.
Yes. This creature, though have coastal striguli, it's not a moth. It belongs to order Hemiptera. I mean, it's a bug. So it's actually belongs to genus Elasmoscelis, belonging to order Hemiptera. So it's not moth. But interesting thing is, whenever I observed this insect at field back in 2020, I had to touch it to be sure whether it contains scales or not. So I think that it is successful in deceiving me. And I think that some other members of Hemiptera order, like some members belonging to family Rhycanidae, which are plant hoppers, they also appear to be like moths, or they also have this characteristic, which is coastal striguli. I think that there's something something common between them. Possibly this characteristic is part of their defense mechanism, or possibly it's a mimicry, or possibly it's a mere coincidence, whatever it is. Now question is how to differentiate subfamily Olethriotini from subfamily Clidanotini. Let's try to understand this difficult part. So it actually needs very well understanding, and it's a very time-long process, I think, to identify these two subfamilies, because the characteristics which differentiate these two subfamilies are so minute and so subtle that we often need to examine that image with better resolution, then following their characteristics lying on their head and labial pulp, I think that we can differentiate these two subfamilies. So in case of Olethriotini, their heads usually have rough scales on vertex. You are seeing here, which is very easy to see in the second image. You are seeing here that their heads have rough scale on their vertex. You are seeing here, and you are seeing in this image as well. And their labial pulps can be ascending, descending, or porate. You are seeing that this one has labial pulp which is ascending, while this one has labial pulp which is somewhat porate. Now question is, what is the characteristic of their labial pulp which can be used to identify this subfamily? And that is, their labial pulps are usually tufted or scaled or contains hairs. You are seeing here that in this moth, which has ascending labial pulp, contains tufts or scales on its labial pulp. I think it's visible here. And in the second image, you are seeing that it's easily recognizable that their labial pulps are tufted or contains hairs or scales. So following this character, I think if we will find any member having history and labial pulps usually tufted or scaly, then it can be pretty sure that it belongs to subfamily Olethriutinii. However, there are some exceptions. But in most cases, this characteristic can be used to separate subfamily Olethriutinii from Clidanotinii. Now, next, let's consider subfamily Clidanotinii. Okay, in case of their labial pulp, they can also be ascending or porate. You are seeing here that in this image, we are seeing a Clidanotinii subfamily member which has ascending labial pulp. And in the second image, we are seeing another moth belonging to this subfamily Clidanotinii which has porate labial pulp. But what's their characteristic on their labial pulp? And that is their labial pulps are usually smooth and not tufted as in Olethriutinii. You are seeing here that in this case, the labial pulps is not tufted as in Olethriutinii. And in the second case, you are seeing that their labial pulps is porate but is smooth, not tufted as Olethriutinii. So considering on the identifying or at least get an idea that how to separate subfamily Olethriutinii from subfamily Clidanotinii. Now, next family, which is family Immidae, which belongs to superfamily Immoidea. The family name is derived from the word imma, which means immaculate or uniformly colored without spots or marking. The name is like this possibly because the type genus and type species belonging to this family, which is Imma rugosalis, which was described by Walker, while discussing on its characteristic, he wrote in this that wings with the lines wholly opposite. Possibly because of this obsolete lines on wings, which means that uniformly colored wings, they are so named, I think. Now, how to identify these family members? Okay, the problem is moths belonging to this family often appear to be as family Tortricidae and subfamily Tortricini therein. You are seeing here that this moth is very much similar to Tortricini subfamily members. And one of the most important thing to be noted is that still now we don't understand this family we don't have a good understanding of this family because this family is taxonomically unresolved and we lack any good data on this family. So we need some workers to resolve this problem. I mean to revise this family taxonomically. Now, how to identify these family members? Well, they are usually small to medium-sized moths with approximately 14 to 42 millimeter wing size, and their heads are usually smoothly scaled. You are seeing here, and their labial pulps are stout and upcurved. You are seeing here. I think it's visible from this image that their labial pulps is upcurved, though this is not like Gelechioidea. And some times when they rest, usually keeps their antennae backward, just like crambids, and their eyes often tends to be of darker or cyan colored. You are seeing here. And in Bangladesh, we have two species belonging to this family, and that is Imma mylias, which is widely distributed in South Asia. This is the moth. It also occurs in India. And second one is this species, which is still unidentified. We have not been successful in identifying this moth. We assume that it belongs to genus either Imma or either genus Mocha, but we need confirmation that which genus actually it belongs to. And this was all I made for today's discussion, and grateful to Moths of India team and to everyone who has listened to my discussion for this long time. And I am sorry to everyone, especially to native speakers of English because of my poor English skill. And I would like to give a special thank to my partner, to my friend Saima, because without his assistance, without her assistance, it was not possible for me to gather these informations together. So I am very grateful to her. And thank you everyone. And thanks to Moths of India team.
Thanks.